Hello, my dear students. Welcome back to another learning lessons in my Physio-Pulse channels. Today we learn about glycogen synthesis, that is glycogenesis, in a simple, easy to understand way. You will find clear explanations, diagram, and important concepts here. Before we begin, if you find the video helpful, please like, share, and subscribe to support the channel. Now let's understand about the glycogen synthesis, that is glycogenesis. First of all, the definition of glycogenesis. Glycogenesis is the process in which excess glucose is converted into the glycogen for storage, mainly in the liver and as well as the skeletal muscles. So, synthesis of glycogen from glucose is called the glycogenesis, and it takes place in cytosol and requires ATP and UTP urinary triphosphates besides the glucose. Now, let's to see step by step how glycogen is synthesized from glucose. First of all, the synthesis of the UDP-Glucose. The enzyme hexokinase in the muscle or glucokinase in case of liver, they convert the glucose into the glucose 6-phosphates. So glucose is converted to glucose 6-phosphate with the help of the phosphoglucomutase, and this enzyme catalyzes the conversion of glucose 6-phosphate to glucose 1-phosphate. Uridine diphosphate glucose UDP glucose is synthesized from glucose 1-phosphate to glucose 1-phosphate and UTP by the UDP glucose phosphorylase. So this is the first step, synthesis of UDP glucose. In the subsequent steps, if you see the requirement of primer to initiate the glycogenesis, a small fragment of pre-existing glycogen must act as a primer to initiate the glycogen synthesis. It is recently found that the absence of glycogen primer, a specific protein, namely glycogenin, which can accept the glucose from UDP glucose. The hydroxyl group of the amino acid tyrosine of the glycogenin is the site at which the initial glucose unit is attached. The enzyme glycogen initiator synthase transfers the first molecule of the glucose to the glycogenin. Then the glycogenin itself takes up to a few glucose residues to form a fragment of primer which serves as an acceptor for the rest of the glucose molecule. So in this step, we want to see that the primer is also formed from the glucose fragments. Now, the third step, the glycogen synthesis by glycogen synthase enzyme. Glycogen synthase is responsible for the formation of 1,4-glycosidic linkage, and this enzyme transfers the glucose from UDP glucose to the non-reducing end of the glycogen to form the alpha 1,4 linkage. Now, this formation of branches in glycogen is that can help glycogen synthase enzyme, which catalyze the synthesis for the linear unbranched molecule with the alpha 1,4 glycosidic linkage. Then glycogen, however, it is a branch tree-like structures, and the formation of the branches is brought about the action of the branching enzyme that is called glycosyl alpha 4,6 transferase enzyme or amylo alpha 1,4, and that is 1,4,2,1,6 transglucosidases. And this enzyme transfers small fragments of 5 to 8 glucose residues from the non-reducing ends of the glycogen chain, that is 1,4 linkage, to another glucose residues which is 1,6 linkage of glycogen. This leads to the formation of a new non-reducing ends besides the existing one. So glycogen is further elongated and branched respectively by the enzyme glycogen synthase and glycogen and glucosyl 4,6 transferase. So the overall reaction of glycose synthesis for the addition of each glucose residues is that glucose whole to the power n plus glucose plus 2 ATP is equal to glucose whole to the power n plus 1 plus 2 ATP plus PI. Of the 2 ATP utilized here, and 1 is required for the phosphorylation of glucose, while the other is needed for the conversion of UDP to a UTP molecule. So this is all about the steps of the glycogen synthesis. The flow chart of the glycogen synthesis or glycogenesis is from glucose to glucose 6-phosphates with the help of glucokinase enzyme, then glucose 6-phosphates to glucose 1-phosphate with the help of phosphoglucomutase enzyme, then glucose 1-phosphate which will convert to form the glycogen primer. First of all, it will form the UDP glucose with the help of UDP glucose phosphorylase enzyme. then it will convert to form the glycogen primer. Now this primer, after the branching and continuously long chain or branching enzymes alpha 1,4, alpha 1,6 glycosidic linkage, it will form the glycogen. So this is the steps of the glycogen synthesis. Now if you see the exactly the significance of the glycogenesis, that is, it can store excess glucose for the future energy needs, number one. And secondly, it maintains the blood glucose levels, liver glycogen. And number third is it provides a rapid energy source during the muscle activity, that is the muscle glycogen. So we have already completed the steps and the significance of the glycogenesis. So if you find this video helpful, please like the video, subscribe to the channel and turn on the notification bell so you do not miss any other biological lessons. Thank you for watching and see you in the next classes. Thank you.